All right. Can everybody see my screen? Awesome. All right. Well, hello, everyone, and welcome to our third online cell modeling software workshop organized by the Center for Reproducible Biomedical Modeling. My name is Veronica Porubsky, and it's my pleasure to present today's workshop on model validation, along with my co-organizers, Sam and Yosef, who you may have met in our previous two tutorials. The main motivation for this workshop is to better understand biological systems using computational tools. So modeling biological systems with mathematical and computational approaches allows us to test hypotheses that help us better understand the system, design new systems guided by underlying principles, or even adapt existing systems. And we can also, through modeling, be able to predict the behavior of biological systems in order to develop various applications, such as medical interventions, for example. As a community, we're still working through some of the challenges that are associated with the modeling workflow, and we hope that these workshops can bring together experts who will be able to present practical tutorials on using modeling software they've developed or tools associated with the modeling workflow, and then provide suggestions for how we can address some of the greater challenges in biological modeling. So today's workshop will focus on the challenge of model validation, and we also invite you to participate in our following sessions on data aggregation and model simulation, and you can also view our previous workshops on parameter estimation and modeling languages in our archive. So just to go through the agenda for today, I'll provide a short introduction on model validation, and then we'll move on to two 20-minute tutorials on model validation platforms, and conclude with a group discussion. Our main goals for this workshop are really to be able to identify common challenges that we've experienced throughout the modeling process, to share our experiences with model validation, our problems and solutions, and brainstorm new solutions collectively, and ultimately build a collaborative community of modelers. During the discussion, all comments and polite debate are welcome, so we really want this workshop to be useful to you, so please jump in if you have any comments. We'd appreciate if you do mute your microphones when you're not talking, just so that we can avoid any background noise. All right, so today, as I said, our challenge is essentially to evaluate the quality and the robustness of biological models. We know that our model is never going to be the truth, but is it accurate enough to be useful? Is it robust enough to generalize to new data and provide some insight into the system that we're trying to study? So by comparing our model predictions to known experimental data, we can essentially compute a goodness of fit measure, and that allows us to show that the model's predictions are statistically similar to the biological data that we have on hand. And ultimately, this will help us evaluate the quality of our model. But still, no matter how well our model fits data, we can never prove that our model is correct, but we can potentially evaluate our model's ability to predict multiple system phenotypes and improve our confidence in our model and demonstrate that our model is robust. So for the challenge of evaluating robustness and model quality, we have a process called model validation, which involves comparing synthetic model predictions to experimental data in order to evaluate whether or not the model is a good and robust representation of the system. So for example, in this model by Carr et al., published in 2012, the model was trained to predict some of the available experimental data, such as growth dynamics over the course of the cell cycle, which is shown in B of this figure. But it was then validated by predicting completely independent data that represents multiple different system phenotypes. For example, the metabolic fluxes of the organism, as shown in E. And while system-wide testing of the model helps ensure that the model adequately predicts these biological phenotypes, it's also helpful to implement a unit testing framework or a series of tests that ensure individual components added to the model function as expected, especially in the case of large models like this whole cell model. And to do both system and unit tests, we have biological model validation platforms, which can check the quality of biological models. So of the biological model validation platforms that are available, we typically choose them in order to address a particular biological context, or in other words, we choose our validation platform in a problem-specific manner. And most platforms can be broadly classified into domain-agnostic or domain-specific categorizations. And in this case, domain-agnostic would refer to frameworks that are adaptable to many biological contexts, and therefore the tests that are written for these contexts can be done as necessary, whereas domain-specific platforms typically provide a series of tests that are uniquely suited to a very particular biological context, which helps them support automatic testing. These platforms perform a variety of tasks. For example, they might provide automatic model checking to ensure that the model meets many specifications that are expected for your system. They also may implement test-driven development, in which tests are written in advance to ensure that whatever defined inputs you have for your system, you are producing the expected outputs in the model as it's constructed. Additionally, these platforms can provide system-wide or unit testing of the model and simulation programs, as I explained before. And ultimately, these platforms provide an opportunity for biological modeling to implement the rigorous validation processes that are actually very standard practice in software development, which can aid greatly in reproducibility and reuse of models. So to discuss two of these model validation platforms, we welcome Dr. Rick Gerkin and Dr. Christian Lieven. For our first tutorial, we're going to hear from Dr. Gerkin, who's an associate professor in the School of Life Sciences and co-director for the Laboratory for Informatics and Computation in Open Neuroscience at Arizona State University. Today, he'll present SciUnit, which is a Pythonic framework for model validation. So welcome, Dr. Gerkin. You are muted.
Oh, thanks, Veronica, for introducing me, and for those introductory remarks. It gives me some confidence that the kinds of things that that I have been thinking about are also the kinds of things that you and hopefully the rest of the group have been thinking about. I'm going to get full screen in just a minute here. Okay, can you see my screen and my first slide? Okay, great. Okay, so I'm going to talk about SciUnit. SciUnit is an example of one of the domain-agnostic frameworks that Veronica mentioned in the introduction. The basic idea is that you don't have to have any specific application to be using SciUnit. It can be used for anything in all of science, not even just biology. And it's a framework that includes a lot of the tools and parts that you would need to make model validation work in general, broadly, and you would then build upon that domain knowledge. So I'm going to tell you a little about how SciUnit works. So it's a framework for validating scientific models. It's based on, like Veronica talked about, unit tests. We think of a unit test as something that a piece of code has to do to be considered functional. A unit test would be usually a sort of a small test, a very specific thing that the code has to do. And so SciUnit just extends the idea of unit testing from generic computer programs to scientific models. And the reason we use unit tests is from the field of software development, we've learned that as programs get more and more complex, the overhead of writing unit tests really just scales fairly linearly. But the benefit you get from unit tests in making sure that your programs work, that you don't have regressions when your programs change or when your model changes, and making sure you don't have catastrophic problems, really increases much faster than that. So it's a worthwhile thing to do when you're writing something that is intended to cover a lot of ground to do unit testing. So imagine we represent the validity of a model. How valid is the model that someone has built by the collection of unit tests that it passes? All things being equal, which is a lot to ask, but all things being equal, model that passes more unit tests, that especially when the unit tests are driven by some sort of experimental data or observations, the more they pass, the better. So SciUnit is a Python package. So if you are following along on the link, I think Veronica provided a link in the chat window to basically this set of slides as a Colab notebook. So I'm presenting here as a Rise slideshow. It's a way of turning a Jupyter notebook into a set of slides. So the notebook that she linked to is this set of slides, but as a notebook in Colab instead of as slides. So what you'll be looking at will, with one exception, be more or less identical to what I'm presenting. Okay. So if you were to get to this line here, you'd be able to pip install SciUnit. It's a Python package, so you're running in Colab, so you're running Python, and you should be able to import it, and everything should work with no problems as long as you click through Google's security warnings and so forth. So here's an example just to illustrate what my goals are with SciUnit and with the related project of SciDash, which we'll talk about a little bit. So here's a very simplified example of the history of cosmology as you may have learned it in high school or maybe even before that. Okay, so imagine that there are these two classes of people, modelers and experimentalists. And so you can think of the modelers as people who developed theories about how the planets move about their orbits, and experimentalists as people who provided data that would constrain those models. And imagine that every one of those experimentalists developed a unit test or a suite of unit tests to test those models. So you start with ancient Babylonians just making sort of good but not especially detailed observations about the motions of celestial objects. Tycho Brahe made much more detailed ones. Galileo, who had access to telescope technology, could actually look at moons of specific planets like Jupiter. And Leverrier, who could look at perihelion precession of individual planets, which is going to challenge a lot of these theories. And so you could look at the different modelers and sort of temporal sequence and ask which of their models, which tests driven by these experimental data could the models pass. So Ptolemy's model and the Copernican heliocentric model are actually about equally good, and for that matter, so is Kepler's model. It's a more correct model. It's more grounded in physics, but in terms of accounting for the data, they actually do about equally well. Newton's model does significantly better because now that we've incorporated gravity, you can understand things like the orbits of moons. And then Einstein in general relativity does better still because it can account for things like perihelion precession that can only be understood in terms of general relativity. So you could look at this and say this is a summary of the field. If you were to enter the field in after the advent of Einstein, ask which model should I pick? You might say, well, in terms of accounting for all the experimental data, the Einstein model looks pretty good. You have some other trade-offs here where in terms of model, say the runtime of the model or model complexity or how easy is it to teach your undergraduate student and so on. And you might say, well, maybe Newton's model is pretty good too. I could use that. But you could use this to help make your decision, right? So it'd be nice to be able to have a scoreboard like this for every field or for every subfield or just for results in your lab. You might have a particular set of experimental data and a competing set of models. You'd be able to look. You'd like to be able to look at a scoreboard like this and maybe click through it and find out different things about the particular tests that were run, the data associated with those tests, how those tests were scored, and so on. Okay. So the goals of model validation, we want to generate at least one unit test for each experimental datum or a stylized fact about the data. It doesn't have to be fitting the exact data points themselves, but it should be some feature of the data that you want to be able to predict. You want to be able to execute the test against all the models that are capable of taking them, recognizing some models can't take some tests because some models aren't designed, even in principle, to account for certain kinds of data. So as an example, in neuroscience, people make models of cells, of action potentials in cells that are entirely point process statistical models. They account for when action potentials occur, but nothing about the dynamics of the action potential itself. So if you have a piece of data that tries to account for the exact membrane potential waveform of an action potential, and that's your data, a model that says just when the action potential occurs is not even in principle capable of taking such a test. And you'd like to programmatically be able to display those results and also understand things that are beyond pass-fail, things about individual scores, artifacts from the tests, like things that are produced in the course of taking the test, etc. Okay, so the workflow basically will look something like this, right? So here's an example of some sort of pseudocode in Python. This particular experimental cell is actually just a markdown cell in the notebook. It's not something you can execute because this is just a high-level representation, not something that I'm intending for people to run. But the idea would be that each of the tests would be somewhere in some module that contains tests. Models would be somewhere in some module that contains models. And essentially, you'd evaluate one test against one model in a very Pythonic way by calling them a method like judge that belongs to the test on the model, and that would return a score. So let's see basically how we get to that point. So the first question is, how does a test know how to test a model? The general problem is you can have many different kinds of tests, which are based on many different kinds of data, which represent different features of the experimental system. And how does it know, how would the test built on that data know what to do to the model in order to coax out a prediction from the model about that data? There's not in general something the test would be able to know, right? Models can be implemented in an infinite number of ways, and there's no way for one test to know how to do them all. So that's really the fundamental problem, I think, in model validation has to be solved first. Otherwise, the model validation is sort of a order of number of tests times number of models complexity problem. And we want to reduce this to something more like order of number of tests, order of number of models. We don't want to have to multiply both of those together. We're going to have a lot of work to do in order to match up all our tests to all our models. So how do we solve that? And SciUnit solves that through guarantees that models provide to tests called capabilities. So what is a capability? A capability you can think of as the way that the test interacts with the model. So the basic idea is that a test lists certain capabilities that are required in order to take the test. And a model implements those capabilities. The test does not have to know anything about how the model implements those capabilities. The test doesn't have to know that the model, for example, uses a certain simulator, or that a model does solves something as a prediction problem rather than a full dynamical system, or that the model uses one programming language versus another even, right? In principle, you could do this, make this very abstract. All the test requires of the model is that the model have a method that matches the signature of a capability. So what's that going to look like? You can look at the code if you'd like for capabilities on GitHub, but I think if I show this example relevant to the Cosmology case, it'll be a little bit more clear. Okay, so here is a big code block, okay? And what I'm doing here is I'm defining a class in Python, okay, called produces orbital position, okay? So this is a capability. It's a subclass of SciUnit's base capability class. And what this capability is it's a class containing several methods, which are signatures of things that a test requiring this capability might ask the model to do. It might ask the model to get a position of something at a particular time and return a tuple of points corresponding to polar coordinates in space of where this celestial object is. It might ask the model to give a period of an orbit, which is going to return some floating value, which is in time, which is a period of an orbit. It might ask the model to return an eccentricity of an orbit, and so on. So here I've defined these, and I've said what the signatures are. And in Python 3.6 or 3.7, you can use this syntax here, which is sort of like static typing, even though Python's a dynamically typed language. It doesn't actually enforce anything, but it does give you clues about what the signature of the method is. What are the arguments types and what are the return value types. Notice that all of these are not implemented. Okay, say raise not implemented error. What does that mean? It means what the capability is saying is that the model, in order to implement this capability, it must take a value like this, and it must return a value like this. How the model does it is up to the model, right? There's no requirement for the implementation. The model can do it in any way at all. It can just draw random numbers. It can solve a differential equation. It can do a machine learning technique. It can do whatever it wants. There are also some methods that are implemented. Why is this method implemented and the others are not? Well, this method doesn't actually require knowing anything about the model. All it's going to do is call other methods that are already implemented and then do some basic math. So for example, if this method, which is unimplemented, is going to return a value in polar coordinates, but we would really want it to value in Cartesian coordinates, that's a simple enough transformation. We don't have to know anything about how the model works to do that. So that can be implemented in advance. Okay. So all capabilities, which are the glue that binds the test and the model, would look something like this. This is a simplified version for this toy example of cosmology. The real code would be probably significantly possibly something more complex, especially implementation, but this is what I'm showing here just to illustrate what a capability looks like. Okay. So SciUnit and the domain-specific libraries that build upon it, which I'll mention briefly a little bit later, can define their own capabilities, okay? And which means they have code blocks that look like that. So for example, SciUnit has some extremely generic capabilities, which are essentially used for illustrative purposes like produces number. The model has to implement certain methods in this class, produces number, in order for a model to have that capability. Here I've commented this line out because we don't have NeuronUnit installed, but NeuronUnit is a domain-specific library for the physiology of single neuron models. So it has capabilities like has memory potential, which is going to have methods that have particular signatures that say that the memory potential should have the following form, the following format, this is the way you would get it, and so on. And then a model can implement that however it wants. Okay. So the next thing to do is to define a model class that implements that capability by inheritance. So one great thing about object-oriented languages like Python is that if you want a model to have all these same methods that were in this class that we defined in the 2 slides ago, all we have to do is use inheritance. So so the model will then be a subclass of the capability like this, okay, and it's also a model. Right? So it's a model generically, and it can do all things a model does in general. But also, all the ways that tests will interact with that model come through a capability, which is a subclass of this capability, this class, like produces orbital position. It could be a subclass of many, many capabilities, indicating the model can actually do quite a lot of things. It implements a lot of things. Okay. So here's an example of a model, a very simplistic model that implements this capability. So I'm going to call it Kepler Model, okay? And it's a subclass here of SciUnit model and that capability. So how does it implement each of these things? Well, some of them are implemented very simply. So for example, period is implemented just as a lookup on a dictionary of model parameters, right? Because the model may have been instantiated to have a certain period. It's somewhere, it doesn't even have to have the same name, period. It could be some other way, it could be computed from some properties in this way. Here, I just implement it as just a lookup on a dictionary. I have other capabilities implemented by, say, computing things on other values, right? So if I want to compute the eccentricity of an orbit, I just have to do this simple equation here. Get position, okay, that's a little bit more tricky. It's going to require that we implement we call some other methods here that you can see don't exist in this class, which is what I'm calling it a base class. But basically, here it's satisfied all the requirements. All of these 4 methods that were part of produces orbital position now have implementations. Okay, and so in order to complete the implementation, to be able to have these things, I actually have to know something about Kepler's laws. So here in this second slide, so this model is just an extension of that base model, has all the same methods, but it also implements all the methods that actually do the work of Kepler's laws here. So some of these things are these terms here are all things that come from solving. If you want to solve Kepler's laws to produce orbital position as a function of time, this is basically what it would look like. So for example, Kepler's laws require solving this implicit function or this equation that equals 0, solving that for this symbol large E, which is called the eccentric anomaly. And from there you can compute, that's basically sort of a measure of how far into the orbit a planet or a moon has gone. You go through a few other calculations, and eventually you can get to the distance from the sun and polar coordinates. Okay? And you get a value like this. So now it's implemented all those things. So from there, we should be able to call any of those methods and get, as a function of time, the location of a planet. Okay? So that was a model class, but now we want to have a specific model. So that model class told us nothing about what planet it was, what moon it was, anything else. So if we want to have a specific model class, we could say, okay, let's take Earth, for example. So we can make a model of Earth. And that comes from Kepler Model, so we'll give it a name. I'll give it some facts, some model. So these are parameters that are specific to Earth, okay, to the Earth model as opposed to some other model. And you could argue, well, wait, aren't these data points? Aren't these observed data? And so that brings up the important point that when you have models, there are things that you put into the model, which may come from data, and there's things you want the model to predict. So in this case, the model parameters are things you put into the model. They might come from known physics. They might come from observations that are sort of unquestioned that we take as truth. But they're not part of what we're trying to predict. So here, I'm just putting in these things as things that which are taken to be true, okay? Which they are. So we have now this Earth model. We can do things that Earth model, like make specific predictions. So this Earth model has the method that came from the class, heliocentric distance. So we can call as a function of time, like right now, we can ask how far is the Earth from the sun? And so the model's prediction is that this is this far away from the Earth, or from the sun, okay? I haven't checked to see what that number is right, but you know, if I've implemented Kepler's laws correctly, then it should be fairly close. Okay, but that's just having a model do something. Haven't tested anything yet. So now let's build a test class that we could use to validate that model. So the first question to ask is, what do we want the test to return? Should it return like true, false, does the model pass? Or should it return some other value? Maybe we want to return a z-score, for example, implementing or reflecting agreement between where does the model fall, a deterministic model prediction is a function of some ensemble of data. Or what is the ratio between the model's prediction and the data's prediction, and so on and so forth. So there are many different kinds of scores that are available we could use. We'll just use a Boolean score or pass-fail to keep things simple. So here's a shot, here's an example of what the test class could look like, okay? So we make a test, the position test, which is a subclass of SciUnit.test. So what does this need to do? First, it needs to say, what are the capabilities that are required? What are the ways in which it's going to try to interact with the model? So what it's saying is, by saying that the test has this as a required capability, is that when it interacts with the model, it will only do so by calling methods of the model that are contained in this capability. Furthermore, the score that will be returned will be Boolean score. So the only two other obligate methods of a test, and there are several others that are optional, but the only obligate methods are, one, how do you generate a prediction from the model, which is going to be something you do through the model capabilities. So in this case, we take a time that comes from the test itself. We ask the model to generate a Cartesian coordinate prediction, and then return what that prediction looks like. That prediction will have a signature that looks something like what the observation looks like. So we get to computing a score between the observation and the prediction. It will be something that we can compute in any way that we want. So for example, in this case, I'm just generating a score that's going to be a function of the distance in Cartesian coordinates between the prediction and the observation, okay? So it looks something like this, right? And in Euclidean plane. And then we can say, okay, now we have to have a criteria for when does this consider to pass. Is it passing if it's within a kilometer of the right, if the observation and prediction are within a kilometer of each other, or 10,000 kilometers, or 10 million kilometers? Here I just picked a number, 10,000 kilometers. You know, for the purposes of someone looking out in a telescope, a crude telescope into the sky, that seems like a pretty reasonable. I don't think you expect Kepler himself to make an observation that's within a kilometer or so. And then we can ask, is this passing? Okay, so this is going to take this value, which is true, false, and it's going to turn that into a score object. So this score object is going to contain a bunch of other additional information we can look at after the fact about the score. Okay, so it's going to return a score. Now we get to the payoff. Oh, sorry, not the payoff yet. We might want to have some extra checks. So this is completely optional, but you might want to have for a schema for what the observation has to look like. You might want to impose some units, some other validation checks. You might want to have some options for the test to do additional things. All these things are optional, but they're things you can do in SciUnit to impose some sort of tests to make sure you don't get garbage in, garbage out. Okay, I won't go into this slide in any detail. So the payoff then is that we create a test. So for example, the test, that was the test class, stricter position test. The specific test is instantiated with observed data. So imagine that we observed on March 1st that these were the Cartesian coordinates of the planet, of Earth. Then we could say this is the test, and I've made up this data here. And then we could ask, how well does the model do on this test? So we take this test. We judge this model, so this looks like the pseudocode I had several slides ago, but now it's actually implemented. And we ask, does it pass? Pass, okay? So that's the simplest view you could possibly get, right? You're in a terminal, you score, you get pass or fail. If there's a different score type, you get a number like a z-score or p-value or whatever else. Okay, but we can get additional information. So the score is also an object, it has its own class, and we can get additional information about it that comes from methods like describe, or what was the prediction, the observation, or what was the raw value that was used to compute it, right? So all that information is there. But more importantly, to get to the vision I showed earlier, with it, we had that matrix of modelers, bioexperimentalists. You might want to actually have many tests that are bundled into a test suite. And so this test could be different kinds of tests, or they could be the same test with just different data that comes from different observations. So we can make a new test, which is Earth's position in April, which again, I've just made up fake data here deliberately to make this test fail. Because if I put in the real data, it actually would probably pass because Kepler's model is pretty good. And then I make up a suite of tests, which is this test and this test. and it's going to have a name. And so when I generate that, and I run it, and I run that test suite against the models, it's going to execute both of those tests, and it's going to show my score. So now it's starting to get a little richer here, right? First, it's showing you some progress information. It's also doing some HTML markup. And if you actually, and this HTML markup is context sensitive, SciUnit is context sensitive. So if you're in a notebook, it's going to show you the HTML output. If you're in a shell, it's just going to show you text, not text with HTML tags, just text. Okay. And it gets richer still, right? So if I were to look at this scores and just say scores in the notebook, you'll get this, which looked kind of like the HTML markup that I did by hand in an earlier slide with Kepler and Galileo and so on. Okay, this is from the actual model and test that we made in Python using SciUnit. Then you get richer still because we could expand this to multiple models. We could make a new model that's just like the Kepler model, except it just gives a random orbital angle. Okay, so it takes the original Kepler model and just imposes and it puts a random number here. So I know this one's going to fail because this is totally random. Right? So if I were to make a new model based on the random model class, and then I were to do the testing of it, you'll see a pass and fail from the good model, and you'll see a fail and fail from the bad model, from the random model. And if we were to summarize that, now we have models by tests, okay? And now we have this one passing case and 3 failing cases. Or we could extract just a slice from that matrix, and it would look something like that. I could extract just Earth model or just Earth position test, and it's context sensitive, so it knows when you're trying to extract a model or a test. You can look at just slices of that, or you could generate score matrix, these rich HTML score matrices. Okay, so what are models that can't take certain tests, right? I give this example of models that aren't even capable in principle of doing what the test is asking. So if I can make a new model that is super generic, right? It has some capability, just not the capability we care about. So it clearly can't do any of these orbital calculations, right? It is nothing. So if I ask this model to take the test, it's going to say NA. It's not going to give me error. It's going to say NA. It's a missing score. And if I visualize what that looks like, it looks like an incomplete grade. Okay. So now we can see which models couldn't take which tests. And in general, you might have some models that can take some tests, some models that can take other tests. And as you build this out with models and tests, you can see the whole ensemble altogether. Okay, so to summarize, SciUnit is used in some multi-scale modeling projects that are going on. Human Brain Project is a very large one. It's about 50 billion euros a year in funding. It covers all sorts of different things involving brain simulations. And so SciUnit is embedded in pretty much all of the model validation work that Human Brain Project is doing right now, especially over the last year and going forward. So we work closely with them. OpenWorm as a project, I think Veronica mentioned she may have had Stephen Larson on in a previous seminar. He's the director of the OpenWorm project. And this is a project that's very multi-scale, going down from biophysics of ion channels in C. elegans to network dynamics in the nervous system to animal behavior taken from videos which you can watch on YouTube of worms swimming around in water. Okay. NeuronUnit is a reference implementation, or you can think of it as the most developed implementation built upon SciUnit that currently exists. It applies to neurophysiology. So SciUnit is totally domain agnostic, and I just sketched out what it would look like if we were to implement this for very simple models in cosmology. NeuronUnit is significantly more developed and applies to single neuron models. So what does it do? It gives many types of model classes that correspond to different kinds of models that exist in neuroscience. Test classes for different kinds of tests that you might want to take. There's 50 or 60 different test classes corresponding to different kinds of features and data that you might want to look at when you're recording from neurons. All of different capabilities, which also have implementations using standard model exchange formats. So for example, you might say declare a model to have a certain capability, but rather than have to write that capability yourself, we already have some of the capabilities implemented, assuming that the model uses something like, say, NeuroML, which is a standard model format in neuroscience. Also tools for constructing tests. Rather than just putting in the data by hand, it would be great to be able to construct the test by just using a REST API from some database, pulling in the experimental data, parameterizing the test, and now you have a test that's ready to go. And so you can do this all in sort of one step without doing anything by hand. and then add tools for executing simulations. So some of the capabilities might require, say, running a simulation, integrating some set of ODEs, so the simulators are supported as well. And you can also optimize models against tests. So you wouldn't want to compare two models against some set of tests where one of the models was just someone's publication of an abstract model, but it hadn't actually been optimized to sort of do the job it was supposed to do. So you can do test-driven model optimization and then compare optimized models on these tests. SciDash, Veronica, do I have any time left?
I think it would be good to move on to our second tutorial,
I will move on then. All I'm going to say about SciDash, if you don't want to write code, SciDash essentially gives you all of the above without writing code. It's a website. It's very much in development. I won't, even the link that's there is not up to date. So I will skip that and you can talk to me another time. So I just want to provide some links here for people who want to look more at the projects, and also say thank you to funding agencies. No, SciUnit was not produced by the combined effort of 3 R01s, but they each did contribute a very, very, very small fraction to development time for it. And I'll also thank my collaborators and people in my lab. So thank you very much.
Thank you so much, Dr. Gerkin. We're going to hold off on questions for the time being and move on to our second tutorial, but please keep all those questions in mind. Do you mind stopping the screen share? Awesome. Okay, so for our second tutorial, we're going to welcome Dr. Christian Lieven. Dr. Lieven is a postdoctoral fellow in the Novo Nordisk Foundation Center for Biosustainability at the Technical University of Denmark. And today he's going to present on memote, which is a software tool that allows us to control the quality of genome-scale metabolic models. So welcome, Dr. Lieven.
Thank you very much. Let me see if I can just get the screen sharing going again. Here. And there. All right. Is that working?
Yep.
Yeah, okay. Cool, okay. So yeah, today I'm gonna talk about this very specific test suite that we've built for genome-scale metabolic models. It's called memote, which is short for metabolic model tests. First, I was asked to give a sort of basic introduction to genome-scale metabolic models. I will kind of go through the reconstruction process because I think that illustrates very clearly what the complications might be with genome-scale metabolic models. So first, you would generally start by building a draft reconstruction from an annotated genome from an annotated genome and biochemical databases. This works by pretty much, or this includes only looking at metabolic genes, because they are the only ones that can reliably be annotated and also make sense for the metabolic modeling context. And then from the reactions, we deduce the chemical equations and then couple that also with gene-protein reaction rules, which essentially couples the metabolic genes with the reactions in the model. And from this, you build a very basic matrix that has metabolites in the columns, and in the rows, you have the reactions. And then typically, after this process can now be automated very well. But after that, typically you begin a very time-consuming process of manual curation, because the automatic pipelines, they don't necessarily cover the latest literature or the latest biochemical and some peculiarities that you can only get from data. So here you essentially have to double-check the stoichiometry, the directionality, and the localization of every single reaction and every metabolite in the model. You often have to add transport reactions and spontaneous reactions, which cannot be inferred easily from the genome sequence. And you manually build a biomass reaction. If you want to look at growth, for example, you manually have to kind of account for that. And you have to add boundary reactions such that the the optimization problem can be solved later on. And then typically you convert it, the entire model into a format that can actually be solved. I work with CobraPy, but the Cobra toolbox in MATLAB was kind of the first thing, or used to be the first software that did this. Now there are several frameworks that can do this, but yeah, again, I work in CobraPy. And here, this would be the first time that you could actually do some quality control and testing. So if people didn't convert very often, it's easy to make mistakes and then only later realize that there have been mistakes made. And then on top of just seeing if the model can actually be simulated, you would ideally also like to validate it against experimental data. So you would be comparing measured rates, yields, or gene knockouts and flux distributions with what the model provides. So since everything is interconnected, you have the metabolites involved in reactions, and the genes also connected to proteins and reactions. But you also have so many other things on top, like annotations, which allow people to map data onto your model. But you also have constraints given, let's say you want to look at growth in a certain medium, then you would constrain a few reactions then compared to different other media. You can have several biomass equations depending on which kind of medium you're growing your organism in. And typically in the reconstruction process, with every reaction, you make an assumption. And then, of course, over the course of several years, you can have several versions of the same model. And all of this complexity adds up. It adds, errors kind of creep in at every step in the process. You can have genomes that have not been sequenced with sufficient coverage to include all the metabolic genes. So you might actually be lacking a few pathways that you don't know you're lacking. Then the databases, they might have been built on a reference organism that also doesn't cover that specific flavor of a reaction. So there could be lots of promiscuous reactions that aren't covered. Then the different software tools, they are still not very standardized. So some tools use outdated parsers and exporters. So you lose data there. And then you just have the sheer plain human error in evaluating your work. And then finally, when you publish, the version might be omitted. So a person that looks at this field and is trying to determine whether model A of E. coli is the same as model B of E. coli might not have an easy time to figure out, yeah, which is which, and which has, which is the latest version as well. So our goal with memote was to kind of, on the one hand, provide a singular quantitative score to make comparisons between all these different model versions easier, but also, of course, to reflect the quality guidelines that people before us have already published. So actually these 96 steps of curating and reconstructing a genome-scale metabolic model, they have been published in great detail. And essentially memote puts this into a form that can be automatically applied. So yeah, in essence, we want to minimize the manual burden of doing quality control, borrowing heavily from software development. So I don't think I have to cover this, but I really like this example. Test-driven development essentially works like this bit of pseudocode here for dishwashing. Let's assume you have a bunch of dirty plates, some soap, and a towel. These are just sort of input variables. But then you have your main function, or your main program code consists of several subfunctions. The first one is to take a plate, the second is to wash the plate with soap, the third is to dry it, and then the fourth is to shelve it. And then unit test would essentially focus on each little distinct unit of your program and make certain assertions of whether they still provide the correct output. And the output that they ought to provide, that's predefined. So in essence, what memote does is it provides unit tests for a metabolic model. So instead of your main program here, you have your metabolic model, and we have provided 148 distinct unit tests that look at the SOPs and that look at previous guidelines and community standards to make sure that the model you're building is up to snuff with all of these requirements. Right, so what kind of tests do we provide in memote? We have a core that works with just the model. So this is really just the unit testing part. And then we also provide some automatic functions that also do some validations if you provide data. So you can kind of imagine memote having kind of two halves to it. And this core section is further divided into a part that we calculate a score on and a part that we cannot calculate a score on because several paradigms exist in the community and there's no consensus yet on whether one way is the right way or the other way. So we have, through talking with the community, kind of distilled out things in a model that everyone recognizes as being very important and that people can give a distinct score on, and other parts are more informative. So you would look at the other sections, for example, here, the biomass section, always with a grain of salt. So if you are looking at modeling, for example, a bioreactor, and you're interested in the growth rate, then of course taking a look at the biomass reaction makes sense. But if you're looking at just general flux distributions in what could be an erythrocyte that doesn't, where the growth rate isn't necessarily important, then obviously you would skip all these tests. Right, but we'll get to the actual tests a bit later. First of all, I kind of want to outline the two principal workflows that you can do with memote. First of all, you can use it to peer review or compare your model with other models, or to just get a glimpse at the general quality of the model, which is not easy if you take a model from someone else. It's often very difficult to get it to run. Like usually they're not provided with Jupyter notebooks that actually can train them properly. So memote serves here as just a window into the tangle of reactions and metabolites to see what's actually going on. So this workflow here is just, you take a model, you run memote, and you get an HTML report that you can then browse through at your leisure and figure out exactly what's going on with this model. But then also the second half here is to aid you in reconstructing. So actually provide you with the necessary framework to do automatic testing while you're building a model. So yeah, here you would just provide, you would start with a draft model. memote creates a Git repository for you. Then memote also provides all the necessary hooks and scripts to always run this, to always run itself, essentially, when you make a commit, so when you actually make a change. Every commit you make creates an entry in what we call a history report, and this can inform you to make choices again on how to proceed next with your curation. Right, so now I guess this is the tutorial part. I'm just going to run through this notebook. Of course, you don't have to follow along. You can just wait until I'm done and then, I don't know, use this afterwards. We install memote comfortably with pip. Here in this case, maybe I should mention that I'm installing a special branch because I realized that I couldn't run the current version on Collaboratory. So this is just a quick fix, but it'll be merged soon, and then it should just be as simple as doing pip install memote. For the purpose of this tutorial, we'll be working with a very small model, the E. coli core model, which is essentially just the core carbon metabolism of Escherichia coli. And we'll get it from the big database, which is a genome-scale model repository of very well curated models. So if you're looking for a model, if you're looking for a way of how to build your own model, and you're looking for some good examples, check that out. So we're just going to retrieve that real quick. primarily memote is a command line tool, so you'll be seeing a lot of these exclamation mark and then some command cells here, which is the way of calling the terminal here in the Jupyter notebook. So don't freak out about the exclamation mark. So let's look at the peer review stuff first. The simplest way of starting is to just run memote report snapshot and then E. coli core. And it shouldn't take too long. What we see here, so under the hood, we're using PyTest. So what we're seeing here is the standard PyTest console output. You can look at this, and I guess if you're familiar, or once you've become more familiar with memote, you'll quickly understand what you're looking for here. You see the individual test categories as individual modules here, and each test is either a dot, that's a pass test, or an F, and that is a failed test. And later on, when we look at the report, you'll know what it means to pass or fail a test and what the criteria are for each. So that's already done. The snapshot report is available later down below. I think I'm loading it. So let me just add that there are some additional options that you can put behind the snapshot here. So you can modify the output a little bit. For example, you can make this very brief and terse console print a bit more verbose by just using this long traceback here. So maybe we can just take a quick look at that, because it doesn't take too long to run. And this actually prints also some of the statements that will be in the report later on. So it has this condensed view again, but then for each failed report, for each failed test, it'll post a traceback style error message that may help you to debug what's happening or to take a look at the model. But like I said, it's nicer to look at the HTML report. But here, for example, we see that the test to check for SBO terms, which is a specific type of annotation, that failed. So yeah, and it shows all the reactions that actually lack this this term here. Yeah, okay. It's also possible to rename the report that you generate by just adding the file name option. You can just run individual tests if you're interested in just this one pathway you're building or this one specific issue you're trying to fix. You can skip other tests that may take too long, and you can modify which solver memote is going to use. By default, it's going to use the GLPK solver because Cplex and Urobi are, they are commercial software. So yeah, by default, we'll take the free software here. But if you have those installed, if you do have a license for them, then you can make memote run a bit faster and a bit more numerically precise by using these two other solvers. Right, so let's take a look at the report here. This is the HTML, the snapshot report that I generated before. We can see that it's separated into two halves. On the left side, we see the half that we score, and you can see the score sort of displayed here in these boxes. So this is each of the tests here has a certain output, and it's color coded depending on how well that test is performing. And if we go all the way down, you'll see a calculated total score, which is kind of the weighted sum of the individual scores. And you also see the score per category. So this model is particularly bad in SBO annotations, or it's not annotated, or not all of the components are annotated with SBO, but it's very consistent. So there are almost no issues with balancing or charge balancing or mass balancing, these kind of things. So yeah, you also see that we've applied some waiting for certain tests that we thought were really important based on our literature review. And obviously a model that is not stoichiometrically consistent will always yield wrong predictions. Like a model that is not stoichiometrically consistent will lead to growth without consuming any carbon source, for example. So some of these models, and that's actually this is also what prompted us to build memote, is that we did find models that had been published that were absolutely wrong, and they couldn't have actually yielded reliable predictions. So, but yeah, as you can see, for each test, we have this detailed explanation of how it works and what it looks at and what the criteria are. And for failed tests, we also provide you with all the metabolites or reactions that you should probably look over in a way that you could just copy paste them into Python or programmatically access them to then automatically fix the issues as well. On the right side, we have the specific section that just looks at, it's kind of informative. So this is something that we didn't calculate towards the score. For example, the basic information just tells you exactly how many metabolites are we looking at here, how many reactions are we looking at, how many compartments are there in this model. Different other matrices are covered here. For example, how many metabolites do we think are unique? So how many metabolites over all the compartments are unique, and how many components are in the medium? So what exactly is the model consuming or producing? And yeah, general matrices that might be of more interest to when you compare models. For example, you might want to know, does the model adequately represent the compartments of the organism that I'm modeling, or does it only have one giant compartment and everything's thrown into that? But we also look at things like the energy metabolism. Is there a reaction that simulates the energy required for just maintaining cell structure? Is there a growth-associated maintenance reaction in the biomass reaction? Is there a biomass reaction at all? There's an entire section on just that. And we also look for specific metabolites that are quite prone to having cycles, and we check for cycles. In this case, E. coli doesn't have all of these metabolites, so or this core model doesn't have these metabolites, so they are labeled as skipped, which is the same thing as NA in the previous presentation. Some people in the community were really interested in just the network topology, so we list here some basic matrix calculations. I guess that's that. And of course, because we want you to share these reports and like exchange with your fellow researchers using these reports, we also include the Python version platform and the version of memote, so that everyone can really understand what the provenance of this report was. But I guess you get sort of the general idea. Let's move on to the diff report, which is kind of the same, or essentially the same as the snapshot report, but here you can feed several models. So I'm going to quickly download the model for human erythrocyte, and we're going to just run memote on both of these models and generate one report that puts the results into context right next to each other. It shouldn't take too long. Actually, I won't wait because I've got the report pre-rendered here, so we'll just quickly look at that too. So essentially, like I said before, it's the same thing. You have just an additional column that shows the second model. And here the color coding highlights like the most significantly different cells or like modeling results here. For example, this guy here, one of the models is better annotated with KEGG annotations than the other model. But it also shows you like size differences and highlights the size difference. essentially the same information as before, but yeah, just side by side, which makes it a bit easier if you want to compare several models. And this takes, I've tested it with up to 3, and I'm pretty sure it takes more, and you could just line them up and look at them in comparison. So yeah, this is essentially what you might want to use if you begin to, or like you want to pick a model to work with in your field, and there are several models available, and this is how you could work out which would suit you better. Right. On to the next aspect. So this is the reconstruction process, and I want to quickly walk you through the steps you would need to set yourself up with a memote repository, which is essentially just a Git repository that's both tracked with memote and has some preliminary scripts pre-installed for then taking it to the next level, which is continuous integration. So the commands here are, again, really simple. You would just type memote new. So prerequisite, by the way, for installing memote is that you also have to install or have to have Git installed. Otherwise, memote won't work at all. Like even if you're just using the snapshot functionality, you would want to have Git installed as well. So no, I'm not going to re-download this, and I'm not going to, oh yeah, I do want to reuse the existing version. So memote new essentially takes you through a couple of steps. So you would provide your name, and I also have to, yeah, do that. And so you provide some email, which I could just use the default here, your GitHub username, and some project name. And I'm gonna name this memote tutorial. I did actually have this name before. Okay, I gotta name it exactly like I did here. It essentially asks you the same thing that you would also provide when creating a GitHub repository or Git repository. So it asks you for a project slug, which is what you would then use in the URL, for example, when you go to GitHub. Some sort of short description. Doesn't matter what I provide here. If you just hit enter, it will take whatever is in the brackets here. So it just assumes the default there. And yes, and for the model, we're going to use E. coli core. And the path to the model is that, yeah. Right. So this actually asking for deployment. This is where the data that is generated, so the results of each run of memote, that's where that is put. And it's put on a separate branch right now, because doing this allows us to also display the history report, the report that is generated with each increment of a model change, display that on GitHub pages, which is hosted online. So if you do this, you will always be able to look at your report by just going to a URL, essentially. So it's quite neat, in my opinion. And hit again, hit. And I think it's complaining because it already existed. I thought I had refreshed it before. Maybe I need to do that again.
Hey, Dr. Lieven, we need to wrap up with a presentation. So.
Oh, really? Okay. Yes. Okay. Well, then I'll just skip ahead. So essentially, this would set it up for you. Would send up a repository. I was going to go through a few quirks of this. Maybe I'll do that real quick. So every time you commit, it will generate this YML file for you. And this is a convenience feature that we implemented that allows you to kind of diff model changes a bit more easily on the file level. So it will show you exactly what you have changed in GitHub. So it'll show this. Unfortunately, it's very small. But essentially on the left side, you will see the old version of the file, and on the right side, you'll see the new version of the model. And this is just a convenience format that's less verbose and more easy to see immediately which of the changes or which changes have been made to a model. So if you keep the changes small in small increments, you will always be able to see exactly how your model has changed. And it'll also generate an initial README for you. And I just wanted to point out that when you use this, you might want to change the license, because by default we add a quite permissive license to the repo. And if you then want to take it to the next level and activate continuous integration, you also have to create a Travis account manually. Right now, there's no way for us to create one for you. So those are just a couple of things you have to keep in mind. And maybe one last thing I quickly want to say about memote is that it also allows you to create. Now I've got to scroll down a little bit. Here we go. To provide experimental data in a specific format, and then it would also automatically run like cross-validate your model against experimental data. But I'll just close here now in the interest of time, and I'll point you to the documentation where this is also featured in detail. Thank you very much.
Thank you so much, Dr. Lieven. We are going a little bit long, but if you guys want to stick around for maybe like 10 minutes, we can just go through a few questions related to model validation, like broadly. Let me just share. I just have like a few questions, or quite a few questions here, but let's look down at the like limitations and solutions category. I think these are a little more broadly applicable. And Dr. Gerkin and Dr. Lieven, you probably have a bit more experience to be able to answer some of these questions, but I guess a lot of these questions are dealing with what kind of standardization could we implement for model validation? What kind of platform would we need to make that happen? Or minimum information standards. I guess with memote, you mentioned that there are 96 steps that are necessary for genome-scale metabolic models, so that's kind of a step towards minimum information. But I guess, do you guys have anything to say about standardization of model validation?
I think some of the things people do in model validation in different contexts, in different fields, a lot of it's the same things, right? Like they have to solve half the problems they have to solve are the same problems in every field, and another half, or maybe more than half, are things that are specific to a field, right? So memote looks great, and if I were in that field, I think that's what I would, in doing those kinds of models, I would just use that. If I were in some other field, and I were to say start from the beginning and say, okay, I want to have model validation for my field or my applications, then I would think about building upon something that's domain agnostic, and then you add on the domain specific components to it. So that's what I'd be thinking about. I mean, it's some of the things that people do independently, like over and over again. It's about as silly as if everyone implemented, Gauss-Jordan elimination every time they wanted to solve a set of equations or something, right? Like we have libraries for that. And then similarly for model validation, it would be good if going forward, as people build new model validation tools, people would think about looking and seeing, okay, is there something like SciUnit that could be used for that? So that's where see if SciUnit would be applicable.
Dr. Lieven, did you want to comment on?
What was the question again out of the bullet points here?
I guess I kind of tried to lump in a bunch of them, but I guess maybe looking at like the systems biology or modeling community as a whole, how can we better implement standardization of model validation?
I mean, the biggest issue that we've run in building memote was the fact that people still used a lot of different description formats. So for us, the biggest hurdle was to just, we got so many different genome-scale metabolic models, and they essentially all had the same components, but where to find and how to identify those components was very difficult. So in the end, we went for specifically just supporting the latest SBML like file standard, a file format, because trying to support even all the different tabular formats that people came up with is just impossible for a developer. So I feel like, and even with the SBML format that exists now, there is still no no consistent way of doing a few things. So there's still a couple of camps of people that like to do it in their way, and that still makes stuff very difficult. Starting, for example, also with nomenclature of chemical entities. Like that is probably the biggest issue we have, is identifying specific chemical entities in a model coming from different labs. Yeah.
Awesome. Okay, so essentially we need, well, it would be easiest if people use the standardized formats, and then model validation could just be optimized to address that standard format. I guess another question I had would be, in terms of automating model validation, what level of granularity we need for domain-specific validation? So, like, if we are interested in modeling aspects of the nervous system, how many cell types do we need to implement? For example, like how many types of neurons? Like, would we need to make specific tests for a given type of neuron? Could we make tests that are applicable to the majority of neurons?
So like in NeuronUnit, there are many tests, maybe most tests are applicable to all types of neurons. I mean, maybe there's a few, non-spiking neurons or something where you wouldn't apply certain tests, but that's fine. You can still run them. You just get, incompletes, right? So to automate them, I mean, the one thing that helps is having repositories of data that have a lot of data being continuously produced about those neuron types, having tools for automatically constructing tests from those data, and to an extent possible, having tests for automatically constructing models based on, standard approaches to modeling. Maybe you have a model that's based on, a biophysically detailed Hodgkin-Huxley model of a neuron where you're programmatically downloading the full morphological reconstruction from some database, and on the other end you have a simple leaky integrate and fire neuron, right? And maybe you want to compare models to say, like, what's the model that makes sense for my application, right? Because there's always going to be a trade-off too between runtime complexity or model complexity and how many things it can do, and individuals have to make their own choices about that trade-off. But I think for automating that, part of it is connecting to, having tools to connect to repositories of data, for example. I love the Travis integration you have, Christian, in what you're doing for memote. We have, SciUnit has there's a config file where you can configure things and you can put that in a Travis file, but we haven't developed like an interface for actually creating for creating the Travis file and adding that in.
I can highly recommend Cookie Cutter if you're interested.
Okay. Yeah,
that's a great Python tool.
Great. That sounds great, because having command line tools is important, right? Because sometimes people are already going to have a Python file or whatever with all of their tests or all of their models that are ready to go, or maybe they're in XML or whatever, and basically just want to have like one line you can run. You don't have to open a new notebook. You want something that can be continuous integration, something you can run every single time. And so it's a good thing. It's a good thing to have. I think we both have it. I think yours is somewhat more advanced.
How do you? Sorry if I come up with my own questions here, but Rick, how do you guys tackle the runtime issue with some of your tests or some of the longer running tests?
On Travis, we don't. I mean, for Travis, we don't. We punt on that issue entirely, because you're not going to run a model a simulation that takes like supercomputing time to run and have it be on Travis's free servers, right? So we have model validation that we schedule on HPC. We have other things that we do in some tests that we actually do have running on Travis, and others that we just have running on desktops. For example, after someone's done some work, they make a commit or they run something overnight or whatever.
Yeah, but then you just run like part of the suite test.
Yeah. So we have suites. Yeah. We have like core tests. For some of the suites are grouped into core tests that are like things that have low runtime complexity, and other things that are, you know, like larger scale simulations where not just simulation, but maybe even the calculation of the features could take a long time. Typically, it's the simulation time itself from the neuron models that takes a long time.
Yeah, same for us.
Yeah, so those get scheduled and run as jobs on HPC like overnight or something.
Since we're like getting pretty late, I just want to let Paul Lang ask a question. He had his hand raised, and then we can wrap up after that.
Yeah, okay, thank you very much. So I'm thinking of probably a rather typical situation of biological modeling where you have a set of ODE models. You have perhaps time courses of the species that are in these ODE models, and you might have some knockouts that are associated with a certain phenotype. And what people have done is that they just formulated an objective function based on least squares of the deviation from the time course, and perhaps also added some terms for violation of the phenotypes at given knockouts. And then they just compare the models by comparing the best objective function of, well, after fitting the parameters. So I was now wondering where in this context unit testing would have a place. And also perhaps if it does have a place, what the advantage would be of SciUnit compared to normal Python unit test.
Well, you could think of SciUnit as like a normal Python unit test. What SciUnit has done is give you the framework to not have to rewrite code every time you encounter a specific, you know, a new problem or a new implementation of a class of model, a new kind of model, a new data type, a new type of test. That a lot of that is you know, you shouldn't have to rewrite tests every time you do that. So there are specific examples of what you're talking about, like with Open Worm project. One of the things that they would do is do optimization of models of ion channels, and they're trying to optimize them to match some data that's recorded in a patch clamp experiment. And so if you change the conductance of some ion channel through some drug or through some genetic manipulation, you're going to get a different model that's going to produce a different result. And you're going to see which of these changes has an IV curve, you know, current voltage relation that looks like the data. And so you have an objective function essentially that looks just like that and measures deviation between the model and the experimental IV curve. And you're selecting models, or you're saying the ones pass or giving up the best score to the ones that have the closest match. So what SciUnit is, you know, in the models in the unit tests that have been written in Open Worm, they're written in SciUnit, do the things you describe, but you just get the things. I think you write less code by doing it in SciUnit, but I also think you write more reproducible code, and you can then extend that to another domain and have that also be in SciUnit. So you can look at sort of an ensemble of models and an ensemble of tests, you know, all together, maybe even the entire project, right? The whole C. elegans organism. Here's the state of the C. elegans model from ion channel to behavior. Here's where we're failing. Here's where we're passing. It's not just one domain like, you know, ion channel behavior, but you can extend that to many domains. So especially for multi-scale modeling projects, I think it gives you some savings.
Okay, thank you very much. I think it makes sense if you can separate it out into different scales. Yeah. Then it works very well. Thank you.
All right, well, thank you everyone for attending, and Dr. Gerkin and Dr. Lieven for contributing your tutorials. I posted a link to our cell modeling seminar Google group just in case anybody wants to continue the conversation. I think there are a lot of really valid points that would benefit from discussion from the community. So thank you, everyone.
Thank you for inviting me. It was great.
Everyone else.